Hello, ladies and gentlemen. My name is Adam Weinstein, and today I will be presenting the gram-negative bacilli Pseudomonas aeruginosa and its role in patients with cystic fibrosis. Pseudomonas aeruginosa is one of the primary agents that has led to the increase in nosocomial infections in recent decades. Hospitalized patients with a hereditary disorder of cystic fibrosis are especially susceptible to infections caused by this organism. Throughout this presentation, I will discuss epidemiological information on both the pathogen and disorder, the course of the infection along with symptoms patients often exhibit, means of identifying the causative agent in the lab, and lastly, the forms of treatment that currently exist to help suppress the spread of the infections and improve the quality of life for the patient. Pseudomonas aeruginosa is an opportunistic organism that thrives in environments where moisture accumulates, such as soil and water in nature. Nosocomial settings, particularly hospitals, also provide suitable conditions for them. They are often found on sinks and doorknobs, which allow the pathogen to be spread to sick patients via direct and indirect contact. Immunocompromised patients inhale it from poor ventilation systems. Thus, it is no surprise that it is the most commonly isolated pathogen in patients who have been hospitalized longer than a week. What's especially characteristic of P. aeruginosa is its array of virulence factors that allow it to resist host defenses, inject toxins into the cells, and ward off a slew of antibiotics aimed to kill it. This is primarily important in cystic fibrosis patients, in which the rate of morbidity and mortality is extremely high due to these factors. Cystic fibrosis is the most serious genetic disease in the population it affects the most, Caucasians. 1 in 2,500 babies are born with it. For the baby to have the disorder, both parents must be carriers. But here's the catch. It's estimated that over 10 million Americans carry the gene, and they don't even know they have it. In the disease, there's a defective gene that changes the consistency of mucosal secretions. So this is going to result in a thicker and stickier mucus, aka sputum, which would otherwise be thin and watery in healthy individuals. And over time, this mucus is going to build up in the airways, and it becomes life-threatening when P. aeruginosa is acquired. And this is because, in most cases, the life expectancy and quality of life are significantly reduced if the infection isn't treated early enough. On average, most CF patients don't make it past their mid-30s. And the chart on the right here shows that the statistics are pretty scary. 30% of CF individuals are infected by age 10, 60% by age 24, and 75% by their mid-30s. I've broken down the course of the disease at the time Pseudomonas aeruginosa is acquired into 3 parts with the symptoms that accompany them. In the initial stage, the symptoms that first appear are coughing, wheezing, and phlegm. At this stage, the host is most susceptible to the organism being acquired because the body is failing to clear out the thick mucus. And the thing with this Pseudomonas microbe is that it has these pili that are really good at sticking to the airways and not allowing the body to push it out. And so it's basically found itself a new home to grow in. And a good indication that there's an infection is the bloody mucus. In the end stage, most patients will develop chronic lung infections from the rapid deterioration of their immune system. This will ultimately lead to death due to respiratory failure. When the microbiology department receives a sputum specimen from a cystic fibrosis patient, there are a number of ways to go about identifying that the causative agent is Pseudomonas aeruginosa. Above all other methods, making a gram stain and performing the microscopic examination are one of the most important steps and should be done first to confirm the morphology observed as gram-negative bacilli. Another important method of ID is the culture. The organism is streaked for isolation onto different types of media, incubated, and observed for growth. P. aeruginosa is aerobic and able to grow at both room temperature and 42 degrees Celsius. The presence of mucoid colonies on sheep's blood agar plates is the defining characteristic to cystic fibrosis patients. A grape-like or tortilla-like odor is also a significant feature from this media. Keeping in mind that these are the most frequently isolated non-fermenters, clear colonies will be exhibited on McConkey agar plates. Finally, Mueller-Hinton plates will show a greenish-bluish color across the agar surface. This is due to the production of two pigments, one of which, pyocyanin, comes only from P. aeruginosa. Biochemical tests are often used to further differentiate organisms and hone in on the pathogen. For P. aeruginosa, the oxidase test is quite commonly used. This test detects the presence of cytochrome oxidase, an enzyme which is produced by this organism. A positive reaction can be observed when the reagent comes in contact with the colonies extracted from a culture plate, and a purple or blue color shows up within seconds. This reaction indicates that the organism does in fact produce the enzyme. Antibiotic susceptibility testing using disk diffusion is routinely used to see which antibiotics the organism is particularly susceptible and resistant to so that effective medication can be administered to the patient. When performed in vitro on culture media, zones of inhibition generally indicate that the organism is susceptible. P. aeruginosa is known to be resistant to kanamycin and susceptible to carbenicillin. Unfortunately for cystic fibrosis patients, there is no cure for their disease. Years ago, before medical advancements were where they are today, Pseudomonas infections were a detrimental sign that death was certain, and nothing could be done to slow down the infection process. In the present time, much improvement has been made and much more research is being done. All newborns in America are scanned for cystic fibrosis so that treatment can be administered early before complications arise. Many medical advancements using antibiotics have been made to slow down the infection. Antibiotic inhalation is becoming increasingly popular and effective. The Toby, which uses a nebulizer for inhaling a tobramycin solution, is approved for users over the age of 6. It's increased the life expectancy and quality of life in many patients. Colistin and levofloxacin are other inhaled antibiotics used for treatment. Hopefully for cystic fibrosis patients, their magic bullet will arrive one day and rid those P. aeruginosa infections so that they can live long and healthier lives. Thank you.